right, well, welcome along. Thank you, everyone, for joining us to the Data Science at Stanford seminar. It is my absolute pleasure and privilege to introduce my friend and colleague, Russ Altman, to you this afternoon. He, as we were just pointing out, has more titles than almost any. He is a professor of bioengineering, genetics, medicine, biomedical data science, and by courtesy, computer science. He's also the division chief, as he just reminded me, of systems medicine in the Department of Pediatrics, which is pretty impressive. So that's pretty much his whole bio right there. But since that would not remotely do him justice, I should remind you that he did his bachelor's at Harvard College. He's an MD and PhD from Stanford University and describes himself as a Stanford lifer, which is good news for the rest of us. He was the chair of the Department of Bioengineering for several years and is really a force of nature within the Stanford community. His own research work has focused on the action of drugs, and in particular thinking about structural modeling of proteins and RNA. He moved the computational work into the realm of knowledge bases and has for many years led the world's foremost database of pharmacogenomics called PharmGKB, and it's a real honor that is hosted here at Stanford and has led to many different advances in a variety of areas, not least its impact on personalized medicine, which has been an interest of his in the recent years. And he, in fact, started a company with some illustrious colleagues from Stanford a few years ago called Personalis, which is now at Series C funding and currently present in 10 different countries around the world. A major part of Russ's work has been his legacy of students because he has also run the biomedical informatics training program over the course of the last several years. And many of us, I think, judge our worth in the world by those we have trained and those that come after us. And I think his legacy would be very significant if that were the benchmark on which we were to judge him. None of that even speaks to those who know him well, which is if you look around the room when someone comes in it, I think you can judge the character of the person coming in. And if you look around the room when Russ comes in for a meeting, everybody's mood lightens and they immediately start to smile because they know it's going to be a good meeting full of creative thought and energy. So it's really a great pleasure to welcome Russ.
Thank you very much for an embarrassingly lovely introduction, Ewan. Yeah, so the best advice I got as a child was never be a jack of all trades and master of none. And that's why I have 7 departmental affiliations. I'll leave that to you to do the calculation. But I'm very excited today to talk about the work going on in the lab. We do a lot of stuff. It's all data to us. It's all biomedical data science. And today the data source that I'm going to focus on is text, biological text. And it is important to our work, and I'll tell you why, and I'll tell you some of the interesting things that, of course, people other than me have done, who I will try to give credit throughout the talk. So yes, as Ewan pointed out, we've been building the pharmacogenomics knowledge base, PharmGKB, for actually 15 years now. And it just got renewed, so we'll be going at least to 19 or 20. And it is a resource of human genetic information and how that impacts your response to drugs, human response to drugs. And it is a well-trusted resource, primarily because there is a devoted group of curators, some of whom are sitting generally over there, that read the biomedical literature about genes and drugs and how they interact, and carefully, and I would say lovingly, extract key information about the important relationships that allow us to understand the molecular basis for drug response. That's useful not only for understanding the genetic influences on drug response, it's useful for lots of other things, like understanding how drugs work in the first place, how you may use old drugs for new purposes, how you can understand the toxicity of drugs. But it's incredibly expensive. Not because I pay them so much, but there's a lot of them, and they work very hard, and so human curation of the literature is an intense activity. And so one of the things that we've always been trying to do is build tools, never to replace these curators because they're too good and they're too human, but to at least give them tools to make them more efficient. And that drives a lot of what we do. It's much of pharmacogenetics and pharmacology is about pathways. And so this is for a drug called warfarin, which is the little purple square at the top. These little eggs are all the genes that are involved in how warfarin does its task, which is to anticoagulate a patient, make their blood thinner is the colloquial term for it. It makes it harder to clot, And that is a good thing if you're having problems with clots, for example, heart attacks, strokes, or clots in your legs or in your lungs. So that's how warfarin works, and it's a bunch of genes that interact in pathways. I'm not going to go over the details, but it takes a village of genes to do this, and every one of the elements on this graphic is not random. It's based on a PubMed, which is the database of literature for medicine, a PubMed citation that establishes that that gene should be on this diagram and where it should be on the diagram. Warfarin has a pathway for how it's metabolized by the body. Separate from how it works, the body has to metabolize it and get rid of it eventually. There's a different set of genes, also shown as these eggs. And again, every one of those elements of this picture and the arrows and where they are on the picture is based on a careful analysis of the literature. And then just to show you, and this is a terrible slide, but it's just one little slide that shows that at the end of the day, we really are talking about places in the genome. Like this is the genome is 3 billion bases. There's one position. It's in gene CYP2C9. Remember, you get one base from mom and one base from dad. And so if you got a C and a C from mom and dad, then you may require a higher dose of warfarin compared to the patients for whom the FDA approved the drug. Whereas if you have a TT, you may require a lower dose than what the normal, I don't want to say normal population, what the population that the FDA approved the drug for. And so we have literally thousands of these that can be then used to personalize drug treatment. And if you were to press this button, View Evidence, which I did this morning, that has 66 PubMed articles that support it, every one of which the curators read, extracted the key information, the gene, the drug, the variation, the phenotype. And so a real challenge for us in data science is, can we even come close to getting that kind of performance from computer programs so we can make the curators more efficient, or so we can make their jobs so easy that they spend more time at the beach and less time in Sri Ram? Okay. And just to say, in a paper led by our host today, Ewan, Steve Quake challenged us all a few years ago to take his genome and assess everything we could about his risk for common disease, his risk for rare disease, and his drug response. And we participated in the drug response part, and we had a great time, and it showed us that this pharmacogenetics has a lot to say about a lot of drugs for a typical person. And I won't say any more, but we have a whole research effort in the lab pushing pharmacogenomics to practice. But I want to talk about text, and I'm going to tell a few stories. And the first story is understanding how to augment a network of molecular and cellular data with textual information. So I mentioned PubMed. We're very lucky in medicine. Many of you know this. We have all abstracts of biomedical periodicals are pretty much uniformly available, free of copyright restrictions for analysis. So there's 24 million, probably about 20 million of them have abstracts. And so we have a big amount of text available in the public domain. And in fact, that represents most of our biomedical knowledge, if you think about it. That's where we have our knowledge, and we write textbooks and we do other things, but this is the core data. And we were interested in knowing if we could get computers to read these abstracts and reason over them to make predictions that are novel. In particular, predictions about drug-drug interactions. So I want to, one of the themes today is that we benefit and build on the shoulders of our colleagues in computer science who just do spectacular things. I'm going to mention several computer science faculty today. This particular work rests on the work of Chris Manning in the Computer Science and Linguistics Department, whose group has created something called the Stanford parser. And the Stanford parser is significant for us because it can take a sentence. You might not recognize it, but under this column is a sentence. ABCB1 is a gene. So it says that the ABCB1 C to T polymorphism at position 3435 influences methotrexate sensitivity in rheumatoid arthritis patients. That's a single statement in an abstract somewhere. And if you put that through the Stanford parser, it gives you a parse, but it also gives you something a little bit more useful computationally, which is this thing called a dependency graph, which usually has the key verb at the top. And basically, it's like when you went to grammar school. It has the subject on the left and the object on the right. So the subject is a polymorphism, and indeed you can see a polymorphism of the gene ABCB1. And the object is sensitivity, and in particular methotrexate sensitivity. So even though this is a statistical parser, I'm not going to go through how it works, but it is remarkable because they trained it on, I believe, the Wall Street Journal as its corpus. It was not trained on biomedical text, but we've had a lot of luck getting it to parse biomedical sentences without having to retrain it, which would be a somewhat painful activity. And a lot of our work depends on our ability now to identify the parts of speech. What's the noun? What's the verb? A common approach in natural language analysis has been what we call the bag of words approach. You look at a sentence and you just consider it a bag of these words. And you can get so far with that, but you can't get very far, because especially if you're talking to our curators, because the semantics matter a lot as to what we're going to put into the database. And so we need a higher resolution semantic understanding of the sentences, and the Stanford parser begins to give us that. So in previous work, now 5 years old, we showed that we could identify the gene, identify the drug, and turn this sentence into a kind of standard proposition, which is that a polymorphism in a gene influences the sensitivity to a drug. And in fact, in work that I'll talk about in a few minutes, we also figured out ways to standardize the language. So there are a lot of synonyms for polymorphism. Polymorphism is another word for mutation or genetic variation. Influence has a lot of synonyms, like affects, upregulates, downregulates. Sensitivity has a lot of synonyms. In work that I'll show you in a little while, we figured out ways to find these synonyms. And so in this paper, we showed that we could map generalized text to normalized text, which you see at the far right. And again, I won't go into the details, but that's very powerful, because then we could have sentences that say the same thing in a different way. And by putting it through this pipeline, we get the exact same normalized relationship at the end, which is very useful. It also reduces the language that we have to worry about, because we now have a much smaller world of statements that can be made. And you can see the reference at the bottom. But Beth Percha in the lab, who's here, and this is when she was a youth 3 years ago, took that work and extended it in an interesting way. So first of all, she applied it with some modifications to 170,000 sentences that led to relationships from all the PubMed abstracts that involved 40 key genes. We know about the key genes in pharmacogenomics because of PharmGKB. And this is a somewhat spectacular graph, I hope you agree, very beautiful, where, let me remember, what does it show? Genes are in red, and drugs are in green. And every blue line between the gene and the drug represents one sentence that we've normalized to a relationship. So for those 40 genes, this is their full set of relationships to the drug formulary. And there's a few hundred drugs on this screen. Of course, the relation is kind of captured by that verb that's normalized, and we have some context. Like, you remember there was the word sensitivity was in there, and expression was in there, and that you can kind of roll that up into those context glyphs, so that every one of these arcs is a green to red arc shown in blue. So let's just do a little expansion, a close-up of that thing in the upper right, which is all the sentences that were in these abstracts that relate a gene, a very famous gene, ABCB1. For those of you who know about transporter molecules, this is the P-glycoprotein, or multi-drug resistance. It's very important for establishing resistance, especially to cancer drugs. And it interacts with this other drug, verapamil, which is a calcium channel blocker used in cardiology for a variety of reasons. And you can see at least 4 of the normalized sentences. So they say verapamil metabolism is influenced by mutations or polymorphisms in ABCB1. The drug verapamil is metabolized by ABCB1. It's a substrate. Treatment by verapamil causes repression of ABCB1. So they have a rich relationship. All of those are actually a little bit different and describe a constellation of relationships between these two genes and drugs. So that gives you a sense for what this gives us. But remember, we could have read all of these abstracts and inferred all of this knowledge ourselves. So what we're really interested in is learning something new. And this is where Beth had a very good idea. She said, well, each one of these relates a drug to a gene. Why don't we chain them together? So here's a drug and a gene, and another drug and the same gene. Let's create a path, and let's see if that path can be used to predict things. For example, drug-drug interactions. So let's say that the left side of this graph told us that drug A decreases the levels of gene, and I've color-coded the part of that path that we would use to figure that out. Drug A decreases levels of the gene. The second half of that path tells us that the gene metabolizes drug B. That means that if you take A and B together, you're going to get an unexpected reaction, perhaps, to drug B because the levels of the gene are not what was expected. That's a source of a drug interaction. So Beth took a whole bunch of known drug interactions and created a classifier to figure out which of these paths would be highly suggestive of a drug-drug interaction. And again, I'm not going to go through her machine learning approach, but here's a list of the pairs of drugs that came highly ranked as very likely to be drug-drug interactions. Now, the ones that have a 1 in the second column are ones that we did not use in training, but that we have a gold standard list of drug-drug interactions that are true. So it's very nice to see a bunch of 1s on that list. But really, the discoveries are the zeros. The zeros means this has never before been suggested as a drug-drug interaction, and yet it's getting a score right up there with all of these well-known interactions. So those are worthy of additional study, and I'm going to focus on the first yellow one, which is metoprolol and dextromethorphan, which get a 0 but get a very high score. So metoprolol is a beta blocker. It is used for controlling the heart rate and for controlling blood pressure. And dextromethorphan is a cough medicine. So what's up with that? Well, in 2000, in some journal, somebody wrote a sentence here which I challenge you to try to read. I don't think you will be able to read it, both because it's too small and it's terrible English. However, our drug normalization widget, if you will, came up with a beautiful summary of that sentence, which is metoprolol, the drug, is metabolized by the gene CYP2D6. One of the things I've learned is I far prefer our simplistic summaries over the actual things that are written. So fine, in 2000 we learned, so to speak, that 2D6 is a famous metabolizing enzyme, metabolizes metoprolol. In 2005, in a different journal, there was another sentence, a little bit easier to read, that basically said that 2D6 also metabolizes dextromethorphan. So now we have this idea that 2D6, this gene, metabolizes two different drugs. That came up in Beth's classifier as a very likely scenario for a drug-drug interaction because one drug can saturate the ability of the enzyme to do any more metabolism so that when you add the other drug, it's unable to do its thing on the second drug. It's already saturated, and therefore you get these symptoms of an overdose of one or both drugs. That's the mechanism, and it's actually immediately suggested to a biologist but just by reading those sentences. So that was her prediction, and it wasn't in any of our databases, but we were very gratified that in 2011, 6 years after either of those papers was written, there was a case report of a patient who had a very bad reaction to dextromethorphan who was on a metoprolol drip, which means they were totally saturated with, their CYP2D6 was totally saturated, most likely, by the metoprolol, and then the addition of the dextromethorphan caused a clinical syndrome called myoclonus, which was reportable and a big deal. So that was that's not a proof, except it was a satisfying confidence builder that these predictions really do make sense. So that's the end of that story. Now, I want to get back to this issue of how do we find the synonyms, because there's been a lot of work by others and by us in this area, including by Beth and a new student in the lab, Yu Hao Zhang, and I want to tell you about that because it's very cool. There's this idea of random indexing. The idea is that each word in our language gets a unique random vector. So it's random, but it's unique to that word. And we're going to encode the word in two ways. We're going to encode it by this random vector, but we're going to come up with a second encoding, which is based on the words that tend to surround this word. So we're going to kind of define the context in which this word appears. And then we're going to look for similar context because we assume that words that are synonymous will be very similar in the company they keep, if you will. And then the question is, can we use this to automatically learn synonyms? So, for example, there's this word polymorphism, which if you're not a geneticist, you probably haven't heard, and I told you it kind of means mutation. Here's a whole bunch of sentences that include the word polymorphism. And let me just take you through how we would do this kind of random indexing. And random indexing is one of a class of methods that look at context in order to define words. And really the key idea is you define a word by the company that it keeps. So here's a little toy example where we've given each of these words, gene, diabetic, and patients, we've given each of them a random vector by assigning zeros, ones, and negatives ones, literally randomly. There's no semantic relationship. We just made up those vectors. But then whenever we see the word polymorphism, we add up the surrounding words to the left and to the right. And we do that for all those sentences that I showed you, and that's hundreds of sentences. And that gives us a profile, this very colorful vector, of the word polymorphism, which we can then take and use similarity measures. For example, it's a vector, and so we can calculate the cosine between any two vectors. If two vectors are identical, the cosine of the angle separating them is zero. Well, the angle separating them is 0, and so the cosine is whatever the cosine of 0 is. I can't remember. It's 1. Yeah. And as that angle increases, they become less and less related. So Beth took 494,000 sentences. She implemented this random indexing. She played around with a bunch of parameters. And then she got this nice table, which has some concepts and some roles. So inhibition and suppression, pretty good, right? Those are, for all of these, they are, well, I'm going to call inhibition and suppression a good one. But now look at the next one. Downregulation and upregulation. They are not really synonyms, are they? But you can see why they would share a context. So we have a little bit of work to do there because really precise antonyms may well have similar context. So that's kind of a flag for methodological improvement. I love the next one, incidence and prevalence. If you do not know what they mean, they are synonyms. If you know what they mean, they are definitely not synonyms. And so again, you can be tricked. And so in many ways, it's the negatives that tell us where we need to work. And we're doing some new work, and I'll give you a little sense of it in a moment. And then we have other ones, investigate and examine at the bottom, assess and evaluate. These are all looking pretty good and give you a sense for how we were able to find synonyms with a little bit of manual curation to take out the bad ones. Now, many of you may know in computer science that there's this package available called Tyvek, which will do this and many other things for you. And we have a new student in the lab, Yuhao, been just working for a few months. And he's very intrigued by this idea of random indexing. This is another way to do random indexing where you take the word discovers and you define it based on its surrounding words, much like we did for the random indexing. The difference is you're going to allow some nonlinear relationships between the word and the other words, so it's not purely a linear relationship, which is what we had for the random indexing. There's a functional relationship that allows you to weight certain locations and certain words in a way that I'm not going to discuss. It's very similar to what we were just talking about. So when Yuhao took this very similar technology and gave it etanercept as a query, this is a well-known drug, it's an antibody drug, and said, tell me the other drugs that have a similar context, we got a very credible list. And you're going to have to take, if you're not a physician, you're going to have to take my word for it, a very credible list of drugs that both have similar, others are antibodies, AB is often a key it's something as an antibody, and including other drugs that might not have been antibodies but were involved in the same disease process. So that gave us confidence that this technology really does work, and we can apply it to genes, drugs, and perhaps phenotypes or manifestations of drug response, which are really the stuff that we care about for the PharmGKB. Now, Yuhao brought to our attention, and when I say brought, I mean yesterday at his group meeting, or maybe 2 days ago, that this kind of work has been done, and other people were working on places. And this group, Miklov et al., had published in 2013 this interesting observation that if you take a complex vector space created by so remember, they were all vectors, these random vectors. If you plot those vectors, and here we've projected into 2 dimensions because it's hard to show you multiple dimensions. But in 2 dimensions, they noticed that the relationship of a country to its capital was quite preserved as a vector. In other words, you can literally subtract off a certain vector and get from China to Beijing. A very similar vector connects Russia to Moscow. And so they showed in this paper that basically it implied that the random indexing or the word-to-vec encoding of context preserves some of the semantic relationships of words. And so they proposed that you could, by analogy, find capitals of other countries by taking the vector to Beijing, subtracting the vector of China, adding the vector to Japan, and that vector should be at or near the capital of Japan, which is Tokyo. So fine, good for them. Yuhao wondered, would that work in the space we care about drugs and diseases? So he took 24 million abstracts in PubMed, which I think involved 2 million words that needed to be indexed. And here's a 2D plot of drugs in purple, and it's actually very reassuring that all the drugs are kind of in a space, because of course all drugs should have a similar context compared to other things. Then the diseases in blue are kind of in a cluster, and then proteins were in green and orange and red, and different classes of proteins segregated. So this was all very new but very intriguing. And then he asked a very simple question: will the Japan, Tokyo, China thing work on drugs and diseases? So he said, let's take etanercept, which we know treats arthritis. Let's subtract the disease vector from the drug vector. Let's add back a new drug and see if the remaining vector are diseases that drug treats. So cetuximab is kind of the query, and what you get are a hit of a bunch of cancers, many of which are the targets of cetuximab use. So brand new, hasn't been statistically validated, but really intriguing. The numbers are low. You've seen 0.99s before, and now we're at 0.1. But these are the top one. These are the closest diseases to that endpoint vector that he computed. So a very intriguing suggestion that the semantics of biomedical text will also be preserved in these encodings, and that we'll be able to kind of get some traction from this. That's the end of that story on synonyms and the vector space of concepts, which I'm sure I'll be able to tell you more about in the next couple of years, assuming that Beth and Yuhao get their PhD. The last story, 2 more stories. How are we doing on time? We're doing good. Characterizing the universe of gene-drug interactions. So PharmGKB curators are constantly reading the literature, and they're extracting relationships. And we asked the question, how many relationships are there? And can we write down all the relationships that are possible between a gene and a drug? So we know that the literature should contain all the assertions, and we'd like to know what they all are. And so can we characterize the full set of gene-drug interaction types? We're also interested in every individual interaction, but this is about the types of interactions that are possible. So we have a closed world assumption about what is possible when a gene and a drug interact. So going back to the Stanford parser, which I mentioned, here's a parse. That's a sentence, and here's the parse of it. It's very similar to the one I showed. This is the dependency graph, very similar to the one I showed before. It increases the relationship. There's a gene on the left side. There's a drug on the right side. And this is work that Beth has done as well. And Beth isolates the path in the dependency tree that connects the gene and the drug directly. So we have 3A4 through an AMOD relationship to the word expression, through an N-sub-pass relationship to increased, etc. You can represent this path. We're calling this a dependency path between the drug, the gene, and the drug. And I just want to make it very clear, we're writing it down as a vector. That's one path. Other sentences relating this gene to this drug would have other paths. But people don't use an infinity of language. They use a lot of language, but this project relies on the fact that people tend to use sentences they say the same thing differently, but not in a zillion different ways, just in several different ways. So let me take you through the logic behind this project. Here are 2 sets of relationships, one between atenolol and its target, beta-2 adrenergic receptor, or B2AR, and between lisinopril and the ACE inhibitor, ACE, which is the angiotensin converting enzyme. And here's some sentence fragments that are observed in the wild, and here's the dependency path, similar to what I just showed you at the bottom of the previous slide. Now I want to highlight that They are very different, except that they are joined in 2 and 3 have exactly the same dependency graph. That forms a link. If that link is robust, it suggests that atenolol B2AR share the same semantic relationship that lisinopril and ACE share. And that, in fact, the blue sentences, which go with atenolol, and the green sentences, which go with lisinopril, are just alternative ways of saying the same thing. So there's the blue sentences for atenolol, the green sentences for lisinopril that are unique. The red ones are in common. But that suggests that even though we've never observed it, there might be a sentence that's true that says atenolol is an inhibitor of B2AR, which would be the analogical sentence to number 4, which is talking about a lisinopril. Hopefully that makes sense. Similarly, we've never seen it in the text, but we wouldn't be surprised to see a sentence that says lisinopril, a potent inhibitor of ACE, based on analogy with the blue sentence number 7, which is talking about atenolol. So you create this little cluster of gene-drug pairs that seem to have a similar semantic relationship. Now, I wish it was that simple. Here's 4 more sentences. Atenolol binds B2AR. Great, we're going to say lisinopril binds ACE. Well, in fact, somebody did say it. Great, everything's working. Uh-oh, look at number 10. Dobutamine binds B1AR, new drug. Great, same exact dependency path. We're going to impute all of those colorful sentences above to dobutamine as well. Well, wait a minute. Dobutamine does not inhibit B1AR. So it is not true that all of those sentences above, in fact, they are not relevant to dobutamine, even though the binding relationship is shared. What do we conclude from this? Well, I'm just giving you an example, but if you have robust statistical evidence for all of this, you would say that bind is a more general concept than inhibit, and suggest that there's a hierarchy of relationships where things that inhibit do bind, but things that bind do not necessarily inhibit, which if you're a biologist, you know that is actually totally true. So what do we do with this? In a test calculation, a small one, Beth took all the sentences involving drugs and the gene CYP3A4. These are the sentence dependency paths that she found. She clustered them with a novel clustering that is worthy of a talk itself, which I'm going to totally skip over. It's actually a bi-clustering type algorithm, which means it's simultaneously clustering columns and rows. But for our purposes, I want to point out, I think I can have so these are the actual sentences that were observed. So that first row there's one sentence that relates ciprofloxacin to CYP3A4, and it's a sentence of dependency graph type 2, the second column. The next row is quinidine and 3A4, and quinidine has 2 sentences, column 3 and column 4. There are no sentences in common. We had that one common. But there are other drugs in this cluster that allow us to link the ciprofloxacin and the quinidine sentences because they share with both, and therefore they create a little cluster of the 3 of them together. That's the big idea. And that's what this smoothing suggests, is our ability to borrow information from other pairs to kind of smooth out these relationships. And then what we see is that there's an inhibitory cluster. So these are genes that are inhibited by the drugs. There's a substrate cluster in the bottom. These are drugs that are substrates of the genes. And it all kind of gives us clarity in terms of the different relationships between the genes and the drugs. So that was kind of a confidence builder that allowed Beth to do this spectacular thing, which is to take 50,000 gene-drug pairs, as they are mentioned in PubMed abstracts, and 197,000 types of sentences that they appear in, those paths that I showed you, do this smoothing algorithm that I just described vaguely, and come up with pairs of gene-drug clusters where the semantics of the relationships between those pairs of genes and drugs are shared, like lisinopril, ACE, as well as atenolol, beta-2 adrenergic receptor. And it works great. What we have here are some of the most common sentences that you see in PubMed abstracts. So these are the most common ways of saying what they're saying. So drug, comma, a blank inhibitor. See that all over the place. You see it 1,100 times. And there's the little dependency path representation. Or and then a similar list of different but very similar gene-drug sentences that establish relationships between genes and drugs. And it goes on, and we have examples, and then you have the dependency path that's being used. Oh, no. OK. It also has this cool thing where I showed you those sentences in that example. It tells you what the sentences are. So sentences that say drug is an antibody directed against gene are synonymous because they co-occur in this clustering with D is an antibody targeting G. And yes, those are synonymous sentences. So there's this very satisfying thing that we are, without any training, we are finding sentences that are synonymous just because the corpus tells us. The data just tells us these are equivalent and should be linked. And then I have other examples, and you can see they're all, they're very satisfying. And the ones that I like, for example, the penultimate one, drug blocked gene activation and drug inhibited gene. That's great because, first of all, the writing on the right is much better writing. Like if you're writing a paper, say it that way. Don't say it that way. But they do mean the same thing. And notice no shared words. It's the network of related gene-drug pairs that establishes that they're similar to one another. And then you get this spectacular picture. So what is this? Beth has now plotted the little black lines are all the gene drug, all those 50,000 gene drug pairs clustered by similarity in their semantic relationships. And there's a lot to see here, but let me point out that in the inner circle of, well, not inner circle, actually the second outer circle, you see 25 overall clusters corresponding to the cuts shown in the red line. This is a tree, but we've just drawn it as a circle. There's 25. That means that to the first approximation, Beth and I believe that there are 25 different gene-drug relationships. The segments outside that are subtypes of relationships that we believe occur because we can see structure within those clusters. Now remember we had binds and inhibits, and we said binds was more general, inhibits was specific. That's the kind of phenomenon we think is going on when we look at, for example, 14 turning into 14A and B, and I'll show you some examples in a second. So I got to help decode this a little bit. So the black lines are the gene-drug pairs that have been clustered together. You see the 25 core clusters color-coded. Things that are mentioned in PharmGKB are in orange dots, pointed to by the PharmGKB arrow. There's another database that just talks about drug targets, not the full set of genes that influence drug response, but just their target. That's DrugBank, and that's in blue. So notice that in that cluster there's a lot of DrugBank and there's a lot of PharmGKB. That's because targets can also be important for modulating drug response. So it's a sensible overlap. Whereas in the PharmGKB section on the right, there's a lot of orange, but there isn't much blue. That's a segment of relationship space where targets are not what's going on. It's more about the pharmacogenomic interactions. And then there's these light blue things, these, I don't know what, teal. I don't have a good color vocabulary. These teal things that are the height of which shows the frequency of the co-occurrence in sentences of that gene and drug. And I'm going to ignore that for now. So I want to look at some of these clusters. So the yellow cluster, the yellow out of the 25, if you look at all those sentences, it's pretty clear that this is metabolic and substrate relationships. That is to say, the drug is a substrate of the gene which metabolizes it. And it's very clear looking at those sentences. The blue one over there are inhibitory relationships. This is actually the one that's shared between PharmGKB and DrugBank. When you look at these sentences, it's clearly drug inhibits gene. But when you do the ultrastructure of that inhibition cluster, you see two subsets. You see things that are general inhibition, and then you see things that are actually pharmacological antagonists, which has a specific technical meaning and which have generally similar sentences, but there are specifics to those sentences that make them subcluster separately. So what am I saying here? We get incredibly fine semantic distinctions based on the use of a huge number of sentences to understand how genes and drugs are interacting. I don't think it's shown here, but another one of the clusters made a distinction between relationships of a certain type that were rock solid and that had been known for years. They use a certain language. And then novel things that of the same type that haven't really been proven yet and that are still a little bit iffy. The language there is slightly different, so it clustered them together, but it recognized that there was this subcluster that was like iffy relationships based on, basically perseveration and using a lot of words, like with the subjunctive, saying, I'm not sure if this is really true. So it really is very, I would say, exquisitely sensitive to the semantics of the relationship between the gene and the drug. Here's one that was activation relationships, and that breaks into stimulation and agonist. Again, agonist is a specific pharmacological concept that has, and you can see, just to make it clear, there's the subcluster, and there's the other subcluster that breaks up these two that are all generally within this bigger cluster, which we called, whatever I called it a second ago, activation. And then I have to deliver, there are the 25 relationships. This is my English language summary of looking at a bunch of sentences, or maybe Beth's, I can't remember, probably Beth's. Synthesizes, activates, enzymatically modifies. This is a big deal to us because we would be surprised if there is a 26th relationship. This is based on a lot of sentences parsed and analyzed and clustered. And so, yes, it's possible that there are other relationships, but we think we have captured them. It allows us, for example, to build tools for curators where we say, we're not expecting any relationships other than these and their subdivisions that we showed you. So very interesting, and the kind of thing we now want to do for all entity types that are of interest to us in pharmacogenomics. That's the end of that story, and I think we're good. We'll still have time for questions. I want to tell you the last story, which is the large-scale extraction of pharmacogenomic knowledge from full text. Everything I've been talking about so far has been abstracts, and we love abstracts, but we all know that there's a lot of information in the full text of articles. And thanks to a number of things like policies and whatnot, full text is increasingly becoming available. And as are these very powerful systems, such as one created by Chris Ray in computer science called DeepDive, that are tuned to find entities of interest and relationships of interest in unstructured text. So we asked the question, can we recognize our PharmGKB entities and their relationships in full text, either automatically or semi-automatically? This is a big deal because we know that the curators can't do their job just from abstracts. They tell us this loud and clear. They need to read the articles. So we said, fine, then we need to read the articles. And now we have access to enough text to start looking at them. And this is work by Emily Mallory, whose first paper in what I think is going to be a string of papers on this topic just came out. So let me tell you how we use DeepDive. DeepDive is a very general system. We use it like this. So the first thing is you gather all of these text documents, often PDFs or HTML. You find the sentences, you find the tokens, which are like the word roots, and you build the dependency graphs based on the Stanford parser that I showed you. Then you are looking for relationships and entities. And here's a sentence. Entity A interacts with entity B. Now, entity A and B could be entities that I care about, like a gene and a drug. Gene interacts with drug, or gene interacts with gene. In fact, this work is looking for gene-gene interactions. But I could also write a sentence that Russ interacts with Jeannie. Jeannie's my wife. I interact with my wife. So I'm going to pull that, if that was in a PubMed abstract, and there are a lot of interactions, probably not Russ and Jeannie, that are in. But what DeepDive does is it looks for what we call candidate mentions that could be gene-gene interactions. So we don't know what entity A and B are. They could be genes. So it identifies that and says, OK, we have this entity, and there's features surrounding these entities, both in this sentence and perhaps other places farther than the sentence. And we provided in the upper right a small database of true examples of interacting genes. That's key because it's going to use that to try to tune its understanding of when entity A and B are genes based on a few examples and a lot of text that it can look at. So entity A interacts with entity B is a candidate relationship. That's what it is. And we've seen some true candidate relations. It builds a machine learner. I'm not going to go into the details. But it takes these candidate relations, uses the true ones, and comes up with an initial probability for the likelihood that a bunch of sentences are talking about gene-gene interactions. So in this case, we have two genes, BRCA1 and ARNT. Gets a very high probability. That's a pretty confident one. Then there's BRCA1, and TP53. It doesn't look quite as strong. And if I had given it Russ and Jeannie, it would get a very low probability because the words surrounding any text about me and my wife, I can assure you, are not going to be similar to the words surrounding gene-gene interaction discussions. And that's the key insight. So then there's a little bit of an iterative process in step D where you tune this. You say, oh, it's being tricked by certain words are throwing it off, or there are certain examples that are throwing it off. And so you do a little bit of manual tuning until you can really get these probabilities to a point where you're happy with them. And now you have a general system that can take at large scale, and by the way, this is done at a very large scale, something like 36,000 documents, 100,000 documents, full text, really even a leap bigger than the previous work that I was showing you. And it can blow through all of those, and it can find every sentence that is putatively talking about a gene-gene interaction, and it can give you a probability. We love that because we can rank order them, we can figure out what our tolerance for false positives is, get a cutoff, and move forward thinking about what we're going to do with this, perhaps presenting them to the curators for final approval or disapproval. There's no reason in principle why this can also be done for gene-drug interactions, for drug-drug interactions, for gene-phenotype interactions, and that's what's on the plate going forward. Let me give you a sense. I didn't really deliver on what are the features that Deep Dive might use that it learns from those true examples that you've provided it. And I just want to give you a sense. So it might have a dependency path that has gene A interacting with gene B on a dependency path. And if that happens a lot for true gene-gene interactions, that could be a piece of evidence. It might find a prepositional phrase like binding of. It might find a verb that's frequently on the dependency path, like the verb interacts. It will look at windows to the left and to the right. All of these things would tend to separate genes from Russ and Jeannie. And that's the goal, is to find genes with high probability and get rid of all the other things that may interact that are not genes. So when we look at this, and we do in the paper that Emily published with gene-gene interactions, there's many features, but you won't. This is kind of a satisfying list of the most powerful features, which is exactly a single verb between the putative genes. It really should say putative genes because we don't know if they're genes yet. But if that verb is bind, I don't really bind Jeannie. I mean, just bear with me. I don't bind Jeannie. I do interact with Jeannie, but I definitely do not regulate Jeannie. Okay, and so basically that's true not only for me and Jeannie, but for all interactions that would not be gene-gene interactions. Eventually, you would say this must be a gene-gene interaction or this cannot be a gene-gene interaction. And again, it's the volume of data that gets you to that certainty. And then this list goes on much longer and gives you a set of rules that then can be applied to general text, biomedical text, to get high fidelity extraction of both the relationship and the entities. And so I think the key thing to point is that this is the most fair evaluation, and we get about 76 precision, 50% recall over a very large corpus of marked up examples. And you can do fun things like looking at, we have the time dates. So the number one question when I talk about text processing is, what if the text is wrong? Right? What if we're extracting all this information, but they just wrote something wrong in the literature? That does happen. What we say is we are just trying to extract what the author said as the primary task. What the author said they really believed on the day they submitted the paper. But you can, as a secondary analysis, you can look at things like temporal trends or the frequency of things being mentioned to make an assessment of whether something is really true or not. So this just shows that for these 10 genes, some of them get very hot and then get very cold and are mentioned sometimes a lot in the literature, and then they're not mentioned ever again. And that might be that's just one example, secondary analysis, that might have us believe or not believe some of these facts that we're extracting. This is just an evocative example to show you that we can use metadata about the sentences to try to do truth determinations. And I'm going to skip over this for time, but these are a bunch of predictions we make. And I'm just going to tell you that in ongoing work, we're continuing to collaborate with Chris to try to do all the tasks critical for PharmGKB, finding genes, drugs, and diseases and symptoms in the literature, establishing their relationships using controlled vocabularies, putting them into databases. The first one I showed you with gene-gene looks like it's working, and we're now moving on to the others. And we're looking at large scale of analysis of all sentences in full text to identify all the human phenotypes relevant to drug response. Very exciting challenges that we are just getting started on. So I'll end up saying that this natural language research is driven by the application needs that we have in doing curation of biomedical data for PharmGKB. We have some really big advantages. I didn't even mention on the slide, but we have these great colleagues in computer science creating very powerful tools. What we bring to that is a laser focus on what we care about, which are genes, drugs, and drug response, which allows us to kind of simplify our use of these tools and really go for it. And we have experts who can tell us, the curators, when our performance is acceptable, when it's not acceptable. And we can make inferences like those drug-drug interactions. And I didn't stress it, but Beth also made predictions about things that should be in PharmGKB based on sentences she found and drug gene sentences that she found that should have been put in PharmGKB. So we do turn this into an inferential activity. So we're excited because in the lab in general, we're combining text analysis with lots of other data analysis. It's the beautiful thing about informatics and data science is you are not tied to a scale. So we think about 3D structure, we think about cell expression, we think about patient medical records, and we think about text, and we bring them together to try to understand drug response. So I want to thank Beth, Emily, Yuhao, who are the graduate students whose work you've seen. Chris is our collaborator, and I want to thank all of our sources of support. And thank the PharmGKB team, which puts together the resource that drives and motivates a lot of this research.
So we have time, I think, for a couple of questions, if you want to raise your hand. Let me ask you, you talked a lot about abstracts and full text of the biomedical literature, but a lot of these sorts of semantic tools have been applied to other information sources in the public domain, Twitter, the internet. What sort of other sources have you looked at, and have you looked at some and discarded them, or? So because again, this provides us some laser focus because the mission of the PharmGKB is to provide scientifically peer-reviewed, good information to the world, and so that gives us an excuse to focus on the published literature. We are well aware that, in fact, I've published about search logs, Twitter feeds, all very relevant to drug response, but in general, not the things you would ever put directly into PharmGKB. They would be more hypothesis generation. So we are very interested in these non-peer-reviewed sources of knowledge, and we are doing work on them, but I wouldn't count that as part of this effort, which is to try to make the curators and make tools, not do anything to the curators, make tools that help the curators be more successful. So we follow that, and so far it's more for hypothesis generation. Question? Back and then one at the front.
I won't be the decider. Yes.
Yeah. Sure. Hi. Great talk, Russ. A couple of technical questions, if I may. The first model, the first story and the last story that you showed us, the first one you had an unsupervised kind of approach where the parser was trained on Wall Street Journal data, and the last one, presumably from the name, I'm guessing, deep dive, so it's a convolutional neural net of sorts, I imagine.
It actually is conditional random fields, I think.
Oh, okay. So there's like higher level stuff that curators take into account when they read the larger paper. So there's, besides just the single semantics of the one sentence that presumably these models can get at, they're basically judging a paper and making a claim that there is a negative relationship here. So this is a negative result, so to speak. So is your team in any way, keeping track of reasons that curators make their decision and keeping a sort of not just a positive control, so to speak, but also negative controls as to why curators discarded or didn't listen to certain papers? So
that's a great question. And the first answer to the question is that we know, in fact, Beth did a little unpublished study that, I can't remember the numbers, but a very small % of the PharmGKB can be recovered if you only look at one sentence. Because guess what? Exactly as you described, the curators not only read papers, they read more than one paper, and they do a lot of inference in their skilled heads to decide that something is true and is worthy of putting into the database. So it is clear, first answer is, it is clear to me that we have to go beyond single sentence analysis in order to even come close to approaching, I think Beth had less than 20%, maybe even single-digit % of PharmGKB can be found in single sentences. So that's the first thing. The second thing is we have not done a formal kind of workflow analysis. I haven't. But I suspect that the curators, through interviews and through examination of they have very elaborate standard operating procedures so that when a new curator comes on board, they can get that person up to speed and how to do this. And I think it's a great idea that looking at that might give us clues as to where to focus our attention at this task of getting better at figuring out what's true.
Yeah, so it might not necessarily be a structured way of collecting information. If you just annotate the reasons they're making the decision, you could probably throw a deep learning network at it and let it figure it out.
Right.
So the second question, sorry, Ewan.
Well, we'll only have time for one more, so make it quick.
Yeah, absolutely.
Okay.
So the first and the second story that you showed, the second, I was expecting you to kind of connect the two. So you were looking at these vectors in Euclidean space based on word contexts around them. But the first story, you had each sentence you constructed pretty much an unrooted tree. So each word, instead of having just a scalar sort of vector around it, each word is surrounded by a tree. And just like you have distances between vectors, you could have distances between trees.
Exactly what we're doing now.
Okay, perfect.
I tried to give it a quick answer.
Yeah, that was very good.
Quick but
You're
true.
good at this. Yeah, we have probably just one last question, I think.
I will hang around for a few minutes. I
Yeah.
apologize.
This is actually a very short question, but do you have precision numbers for both drug-gene and gene-gene interactions? Whatever you extracted, how accurate is it?
Yes, I don't know them, but they are in the papers.
So how high are they? Approximately like 50%, 90%?
So could the people who did the work answer that question quickly?
Yes, the question is on.
Yeah, please. These are the papers, just so.
So the question on the first paper was how high was the precision at extracting certain classes of relationships? We tested two types of relationships in PharmGKB, which are situations where mutations in the gene impact your response to the drug, and we tested drug-target relationships, as Russ mentioned. And so what we were able to do is, using a small set of known relationships, extract other relationships with high accuracy, but the performance depended on the amount of seed pairs that we put into the system. And so in the end, we were ranking test sets accurately. We were ranking, man, it's hard to explain all the all the details of this work and how we evaluated it, but we have that data in the paper, and the precision was very good at the end.
And maybe we could chat. You could chat a little
I'm
bit
sorry,
after
yeah.
this. No problem.
need to-
That's great. It sounds like, so this is actually a great segue into the closing part, which is just to say that this seminar is brought to you from the Data Science at Stanford Forum, which really is, that word is chosen specifically to be non-specific, an entity that really exists to bring together those interested in data science from different schools around the university, hence why our last session, for example, was across the other side of Campus Drive in the engineering school. We are here, and you can go to datascience.stanford.edu to help. That's it, exactly there. To help to make connections. If you're somebody in the School of Medicine who is interested in being connected to engineers who can do this sort of cool stuff, or you're an engineer who has a great tool who thinks, and you think you can revolutionize medicine, if only you had the right data set, then wander across Campus Drive, come to the hospital, come to the School of Medicine, and we can help you do that. So that's the point of data science at Stanford. There's a few of us are listed there. You can contact any of us, and we would be happy. Oh, there's some of the people. And we'd be happy to make connections. The next talk in the series, we're also happy to take suggestions for people you'd like to see talking at the series. The next talk will be by David Lobel from Earth Sciences in January. He's going to be talking about food security for, now I think on my sheet it says 9 billion people, given that I think the Earth doesn't have 9 billion people yet on it. Maybe that's the future. So please thank me. Help me.